Good afternoon. Welcome to Medical. Oh, I hear an echo. Let's see. Well, there we go. Welcome to Medical Grand Rounds. I'm Bob Wachter, Chair of the Department of Medicine. Welcome to our group here in the nursing building and our folks following online. Today we have a very exciting topic and talk that really reflects the breadth and depth of what we do here at UCSF, and particularly today we'll focus on pulmonary disease with a real emphasis on bench to bedside. The topic is epithelial cell reprogramming in pulmonary fibrosis. The talk really is about how we are gaining a greater understanding of what's going on in fibrotic lung disease, which is a fairly common and disabling whole set of lung diseases. Those of us who are generalists try to remember all the initials, and it's hard, but it's important that we understand kind of what's going on and what's underlying the disability that the patients have. And in the last decade or so, we've come to really deeper understanding of what the underlying pathophysiology is, and it's pointing the way toward effective treatments. The speakers really are two of our experts and two world experts in the topic. The first speaker, I think, will be Paul Walters. Paul received his MD from Minnesota, trained in medicine at Colorado, and came to us for his pulmonary fellowship and has stayed on for his faculty career, as often is the case. He's now professor of medicine in the pulmonary division. His primary clinical work is in taking care of patients with interstitial lung disease, and his research program focuses on the role of epithelial cells in promoting fibrosis, and therefore how to understand that and how to stop it. He will be joined by Tien Peng. Tien got his MD at Hopkins, his residency at Columbia, his fellowship at Penn, and then made the move west to UCSF, joining our faculty. And he's also stayed here for his career. And he's really a world-renowned researcher in this topic, with a particular focus on how pulmonary fibrosis happens. He uses a mouse model to understand the pathophysiology and come up with new treatment approaches. Thrilled to do this, and really do believe that this is an area of unique strength at UCSF, and looks forward to hearing it. So Paul is first.
Thanks, Bob. So the talk is titled Bedside to Bench, as opposed to typical bench to bedside. And the reason is that arguably some of the biggest advances in pulmonary fibrosis in the past 10 years have been on the clinical side. And those observations have informed the pathobiology and enabled us to ask better, more informed questions about what the molecular drivers of the disease are. And I'm going to talk mostly about clinical stuff. I'm going to talk briefly about the relationship between aging and lung fibrosis. And I'm going to talk about the genetics of pulmonary fibrosis, which has really been where these major advances have come from. And then I'm going to outline how our understanding of the genetics has informed our understanding of pathobiology, and then a little bit on how we're leveraging all this information in the clinical setting. So the first slide, the first thing that we've learned, let's say in the past 10 years, is that pulmonary fibrosis is an aging-related condition, and that there's a continuum of lung fibrosis in the population. This is illustrated in this first slide, where I take three hypothetical 75-year-old individuals. On the left is the majority of 75-year-old individuals. If you did a chest CT scan, their CT scan is normal. If you did a lung biopsy, the lung is normal. And those individuals lose about 25 to 30 milliliters of lung function per year, starting at the age of 30. So over the course of your lifetime, you can lose up to about a liter and a half of lung function. With the use of CT scans for lung cancer screening and the ready use of them in the emergency rooms for patients with respiratory symptoms, we've recognized that about 5 to 8% of 75-year-old individuals will have very subtle interstitial changes on their chest CT scan. You can probably just barely see them on the CT scan, but they're present. They've done biopsies on a few of these people, and what you find is excess collagen matrix deposition in the lung. Not a lot, but a little bit. We don't know how the lung function in these individuals change, But we do know that if a patient has an ILA, they have worse survival than a age similar individual. Their survival is reduced by about 20%. The other thing we've learned is that if a patient has an ILA and you follow them over the next 5 years, about 8 to 10% of those individuals will develop pathologic lung fibrosis. The canonical disease is idiopathic pulmonary fibrosis. This is what the pathology looks like. That's what the CT scan shows. This occurs in up to 1% of individuals over the age of 75. If a patient has IPF, they lose 300 milliliters of lung function a year, and untreated, the average survival is 3 years. So now I'm going to talk about the genetics of pulmonary fibrosis. Before I do, I just want to I'm going to talk about two things: common variants and rare allele variants. And just to remind everyone, rare allele variants are variants that occur in a gene in less than 1% of the population. You could call them a mutation. They're rare, but they have a big effect. So if you find them in an individual, they have a large effect on whatever you're looking at, in our case, pulmonary fibrosis. Common variants are generally present in more than 5% of the population. They don't have a very big effect at the individual level, but they can have a big effect for risk for pulmonary fibrosis across the population. So the real breakthrough, I think, in our disease came from studying a condition called dyskeratosis congenita. This was first described in the 1950s. It's a syndrome where patients can develop skin hyperpigmentation, funny fingernails, oral leukoplakia. Some individuals get liver cirrhosis. Some individuals get bone marrow failure. Importantly, about 20% of individuals get lung fibrosis. And in 2000, the first gene was linked to this. The gene is called dyskeratosis, DKC1. And importantly, that gene was a gene involved in telomere length maintenance. And subsequently, other genes involved in telomere length maintenance were linked to dyskeratosis congenita, and it was recognized that that disease or syndrome was a disease of telomere dysfunction. After that was discovered, the relationship of telomere dysfunction to pulmonary fibrosis was recognized, and telomere-associated genes became candidate genes as possible causes of lung fibrosis. And the Vanderbilt group had collected more than 70 different families of individuals with pulmonary fibrosis. They sequenced TERT and TERK, two key genes involved in telomere maintenance. And importantly, they linked them to the disease in about 7% of their patients, linking for the first time genes in telomere maintenance to familial pulmonary fibrosis. Following that publication, there was a whole slew of publications over 5 or 6 years looking at candidate genes linked of rare variants in familial cases of pulmonary fibrosis. And the genes linked fell into 3 categories. The genes on the top here are highlighted in red are genes expressed only in the lung epithelium. Genes in black are genes involved in telomere maintenance. And the green gene is a gene involved in spindle fiber formation, DNA formation. So 3 categories of genes are linked to pulmonary fibrosis. At the same time, people were looking at rare variants. 2 groups were looking for common variants. So these are single nucleotide polymorphisms that you find commonly in the population. You need large numbers of patients to link a variant to a disease. The first publication was by David Schwartz's group, and the most highly linked gene was another lung epithelial cell gene, MUC5B, a mucin gene. There's a couple other genes, DSP1 that's expressed in the epithelium, and TERT was also linked to sporadic pulmonary fibrosis. And if you summarize all of the genetic studies, and there's been dozens of them now, it's sort of remarkable because the genes linked to familial forms of pulmonary fibrosis or sporadic pulmonary fibrosis continue to fall in the same three categories. The red genes, again, are epithelial cell associated genes. The black genes here are genes involved in telomere maintenance, and the green genes are genes involved in spindle fiber formation. So the genetic studies really tell us that pulmonary fibrosis is a disease of epithelial cell dysfunction, failure of telomere maintenance, or DNA repair. So how do those genetic studies, how are they informing us about the pathobiology of pulmonary fibrosis? Well, this section of the talk, largely, I'm going to be talking about telomeres. So I just want to remind everyone what a telomere is. That's the DNA sequence at the end of a chromosome. It's a repeating unit of nucleotides, TTA, GGG. It repeats endlessly for thousands of base pairs. And the telomere loops back on itself, and then it's capped with a group of proteins called the sheltering complex. And the purpose of all of this is to maintain or to prevent the end of your chromosome from fragmenting. You can imagine if your end of your chromosome fragments and it fragments enough, it'll start breaking into regions of genes that are involved in protein expression, and it'll mess up a cell. Okay, so the cell expends a lot of energy trying to maintain a normal telomere length. In fact, there's more than 30 proteins that maintain your telomere length. Nevertheless, when a cell divides, all these proteins, the telomerase enzyme complex, is imperfect. And so with every cell division, your telomeres get a little bit shorter. They shorten by about 20 to 30 base pairs. And over time, with cell division, the telomeres get shorter and shorter. And if this telomere becomes critically short, that protective cap of proteins falls off, and it can't protect the end of the chromosome anymore. Now you've got a naked end of DNA, and it activates something called a DNA damage response. And one of two things happen. Either the cell dies through apoptosis, or it undergoes something called senescence reprogramming, which is where the cell can't divide anymore, and it changes its phenotype a little bit. It changes the genes it's expressed and its functions slightly. Okay. Well, is this true in IPF if these genes in telomere genes are really linked to pulmonary fibrosis? There's now been like 5 different studies that have quantified telomere length in the lung epithelial cells of pulmonary fibrosis patients. And in every case, it's been found that the telomeres are short in the type 2 cells of all IPF patients. This little red dot here is a probe that binds to the telomeres. The brightness of the probe correlates to the telomere length. And in all IPF patients, and in patients with non-IPF UIP, which is pathologically similar but driven by a different cause, they have short telomeres in their lung epithelial cells as well. What's the downstream consequence? People have looked at senescence markers in the lung epithelium of IPF. I'm just showing you a little bit of data here, but this has been replicated in multiple different groups. You can stain lung tissue for something called senescence-associated beta-galactosidase. It's not showing up really well, but there's blue cells here in the epithelium. You can stain for P16, another marker for senescence. And these are upregulated in the lung epithelium of IPF patients, telling us that the epithelium in IPF has undergone senescence reprogramming, potentially due to telomere dysfunction. What's another clue that telomere dysfunction is driving pulmonary fibrosis? Well, you can measure telomere lengths in your peripheral blood sort of as a marker of an individual's risk for acquiring short telomeres. And when you do that and you look at survival in patients with IPF, patients with the shortest telomeres in their peripheral blood have the more rapid progression and the worst survival. And it's sort of a dose response. This is the patients with the lowest quartile, the second lowest quartile, et cetera. So they have worse survival. And importantly, they have more rapid loss of lung function as well, telling us that telomere dysfunction may be a molecular driver of the fibrotic process as well. they've done some Mendelian randomization studies looking at genes linked to telomere maintenance in pulmonary fibrosis. And two things were found. This was not a pulmonary fibrosis group. This was just geneticists looking for a relationship of diseases to either long telomeres or short telomeres. Two things were confirmed. One, if you have long telomeres, you have a higher risk of getting cancer, mostly brain cancers. But importantly for this talk, patients with short telomeres had a higher risk of developing pulmonary fibrosis. So telling us that short telomeres are a molecular driver of lung fibrosis. Finally, that's all human data, right? And it's important when you're trying to figure out molecular mechanism to do studies in animal models to see if you can replicate any of the human data. And there's now been 6 different studies that have either caused telomere dysfunction in the lung epithelium or senescence reprogramming of the lung epithelium. And in all cases, those mice develop pulmonary fibrosis. The fibrosis is progressive and the mice die from lung fibrosis. So really telling us that telomere dysfunction in epithelial cells is a molecular driver of pulmonary fibrosis. Everything I've talked about up to this point has been IPF. Once we started looking for this in patients with idiopathic pulmonary fibrosis, the obvious question was, well, do these genetics at all relate to other clinical forms of pulmonary fibrosis? And as Bob mentioned, there's many different clinical forms. I have the acronyms here. The names aren't important. All I want you to see is that there's different sort of visual patterns of the lung fibrosis. And importantly, when we and others found a relationship of telomere dysfunction in the genes related to telomeres in IPF, we started looking in these other diseases. And a sort of a remarkable pattern emerged. And I summarize it on this slide. I mentioned the MUC5B minor allele is the most highly associated gene to the risk of pulmonary fibrosis. The prevalence of that gene is 30 to 38% in IPF. It's similarly prevalent in other forms of lung fibrosis. The one exception actually is scleroderma. It's also a risk factor for ILAs, that sort of intermediate fibrotic individual that I showed earlier. Peripheral blood leukocyte telomere length predicts onset and progression in all forms of lung fibrosis. When they've looked for rare mutations in telomere-associated genes, it's been done in 3 cohorts: IPF, HP, hypersensitivity pneumonitis, and rheumatoid arthritis-associated ILD. You find rare variants in telomere genes at a similar prevalence in all 3 diseases. Common variants, the common IPF variants, yes, they've been found in IPF. They've been looked for in hypersensitivity pneumonitis. They're found at a similar prevalence in that disease. And I showed the short telomeres in type 2 cells and senescent type 2 cells. So it sort of is suggesting that telomere dysfunction, senescence reprogramming may be a molecular driver of all clinical forms of pulmonary fibrosis. Well, then how do we leverage this information in the clinic? An important study was done by Christine Garcia, where she had many families of patients with mutations in telomere-associated genes. And she asked the question, if a patient has a mutation in a telomere-associated gene and lung fibrosis, what does their lung fibrosis look like? And you can see here in the table here, you can have many different clinical forms of pulmonary fibrosis. It sort of flips the question, right, from gene association to the disease to gene association to the pathologic or clinical phenotype. But importantly, no matter what the clinical phenotype was, if a patient had a mutation in one of these telomere-associated genes, their survival and rate of progression of the disease was identical. It didn't matter what it looked like. It really mattered what the molecular driver of the disease was. In the clinic, I mentioned earlier the syndrome dyskeratosis congenita and the clinical features of that. Well, we're using those clinical features in the clinic to try and tease out when we're seeing a new patient with pulmonary fibrosis, whether or not they potentially have a telomeropathy, a mutation in a gene of telomere maintenance. So we ask questions like, did you have gray hair? Did you have onset of gray hair younger than the age of 30? We look for hematologic abnormalities in our patients. Patients with telomere-associated mutations may have an elevated MCV, they may have low cell counts. In some cases, they may have myelodysplastic syndrome. Interestingly, many of our IPF patients will have coexisting liver cirrhosis. This is sort of new information, but there's been 2 studies that have taken a population of IPF patients and with no known liver disease and simply asked the question, do you have any evidence of liver cirrhosis? And you find cirrhosis in about 1/3 of your IPF patients. Again, suggesting a relationship between telomeres and as a driver of these disease processes. Skin and nail findings, they're pretty rare. I think I've only seen it a couple times in clinic, but these other features can be really clue us in that a patient's fibrosis may be due to telomere dysfunction. The other important thing to be aware of is that 15 years ago, before we knew much about IPF, we used to treat them with immunosuppressants. Then Talmadge King organized a randomized trial testing patients with IPF, immunosuppressants or placebo arm, and remarkably, patients that got immunosuppressants did worse. So we were harming patients with IPF with our standard of care at the time. Christine Garcia went back and looked at that population and asked the question, is there a relationship to the harm being caused by these immunosuppressants and telomere length? And what she found was that it was really the patients with short telomeres who got immunosuppressants who had the worst survival, suggesting that immunosuppressants may be harmful in patients with pulmonary fibrosis and short telomeres. Chad Newton picked up on that idea and asked the question in other populations of lung fibrosis, whether there's a relationship between immunosuppressant use and telomere length. Many of these other forms of lung fibrosis, like in autoimmune diseases or hypersensitivity pneumonitis, our standard of care is to treat them with immunosuppressants like prednisone and CellCept. So he asked the question, in those populations where you can find short telomeres up to 1/3 of the time, is there a relationship between outcomes, telomere length, and immunosuppressant use? And what he showed was that patients with those clinical diagnoses of lung fibrosis who had short telomeres had worse survival. So the use of immunosuppressants in that population of patients with short telomeres has worse survival. The way we do these studies is looking at the patients with the shortest telomeres, less than the 10th percentile. But this graph highlights another important point, which is telomere length is a continuum, and the risk is actually a continuum. It's not like there's a magic line in the sand where somebody's risk transitions to high risk. And once you're below about the 40th percentile in telomere length, the risk goes up continuously. So this is my last slide. So how are we doing this? How are we incorporating this stuff into the clinic? This is a work in progress, but more and more we're sequencing patients, especially if they have a strong family history or these features of telomeropathies. We're sequencing them for telomere-associated genes. We're also using peripheral blood leukocyte telomere length testing for 2 reasons. One, it might inform the diagnosis and prognosis, but it may also inform whether or not we're using immunosuppressions in an individual patient with pulmonary fibrosis. And with that, I'll stop, and then Tian will talk more about the basic biology.
Okay. So thank you. Paul really gave a beautiful summary of some of his own work too, of really making these critical clinical observations and how that leads to the basic biology and what we understand about pulmonary fibrosis. So what I wanted to do today for my talk, again, is to maybe flip it a little bit, right? Knowing that we have this bed to bench component that's so important. But then how do we think about things? And once we start to understand them at the bench, how do we flip them back to the bedside? So what I wanted to talk today about is really sort of thinking about, again, the theme that Paul has developed, which is this epithelial dysfunction in fibrosis, but really try to frame this in a therapeutic context and paradigm in terms of how can we approach it to sort of lead to some type of therapeutics in the clinics. So this is my disclosure. This is what our lab studies. So we are a stem cell regenerative biology lab that primarily actually doesn't focus so much on the stem cell, but rather the fibroblasts within the niche and how they orchestrate the tissue response. So for today, what I really want to focus on is the role of fibroblasts. Appearance of one type of epithelia in a place where it is not supposed to be. So in medical school, which many of you have gone to in this room, the most common form that you might have learned about is actually Barrett's. So in this case, in Barrett's, do I have a pointer? I don't have a pointer. Here, Barrett's basically is a clinical condition where you have the normal squamous lined epithelia of the esophagus being replaced by the columnar epithelia of the gastric in a place that's supposed to have esophagus, right? So, and this is due to chronic insult from, you eating your sandwich with hot sauce at Medicine Grand Rounds, you get reflux, and this happens all the time. And lo and behold, you have Barrett's esophagus. So this form of metaplasia is common, but there are actually, this metaplasia is actually a pretty common entity across different types of chronic diseases in different organs. And in the lung, the disease that we're going to talk about, this is actually one of the pathognomonic features of IPF. And in this case, what I'm going to show you, again, in the normal lung, what you have is there are these green cells that are basal cells that normally are only in your conducting airways in the normal lung, and they line so this tube that delivers air down into the alveolar space where you do not have this type of stem cell. But in an IPF lung to the left, as you can see, they are studded with these green airway basal cells in a place where they're not supposed to be. So this form of stem cell metaplasia is actually a really important feature and what leads to the architectural distortions we see in IPF. And this is not just a biomarker for IPF. What we found is that what the others have found is that this process, which actually pathologists have a term for, which is called bronchialization, and it literally means almost like the appearance of these bronchialized cysts within areas of the alveoli, distorting the alveolar space, is that the appearance of these basal cells is actually prognostic. So this group in Germany, André Prass's group, took bronchial alveolar lavages and quantify the basal cell signature within those lavage. And what the group could find is that the amount of basal signature in those lavages is actually predictive of the mortality of the patients. So the more basal cells they have, the more likely they are to die. So the question is, well, having basal cells in your alveoli is bad. Well, how can we, so again, with going along the theme that epithelial cells are drivers of IPF, well, what are some of the things we can do to think about how can we target this as a process? So the two relevant questions first, what I want to understand is what is the origin of these metaplastic basal cells and what is potentially driving that process? And so this is a model that was proposed, and this is what many people believe, that because you have these airway the model, what many people believe, is that there's this migration model where airway stem cells with injury sort of goes into the alveoli to form these bronchialized cysts. And this is a single cell paper from during COVID, where again, in COVID and severe lung injury, you also have the appearance of these metaplastic basal cells within the alveoli. And this is what was proposed without evidence. And I will say that part of the reason for this model is that in the mouse, we do know from very rigorous genetic studies that the metaplastic basal cells that arise in the alveoli do in fact come from the airway. But I will caution you, and I can tell you this because I study the mouse and human, is that mouse are not humans. And this will, I'll get to this point. So their lungs are actually have very different architectural features. So this is the current model. So what, a few years ago, our lab in collaboration with Hal Chapman's group found this very surprising trajectory of stem cell trajectory, where we can take a human alveolar stem cell and we can actually put them in a culture under a specific condition with activated fibroblasts. And what we can do is we observed these alveolar stem cells, like right before our eyes, turning into basal cells through this intermediate steps where there are these alveolar and basal cell features, these cells. So this is just a single cell plot with each dot marking a specific cell type. And so what was also super interesting at the time for us was that some of these alveolar basal intermediate cell types were actually identified in the IPF lung as well, with terms like transitional or basaloid cells. So what we found was that, again, the alveolar stem cells themselves could generate the metaplastic basal cells, suggesting a model with which they arise in situ, right? That is in response to actually some type of abnormal fibroblast signaling as the scar forms in the alveoli that's sort of transforming the alveolar epithelial surface into this sort of abnormal basal cell-like features that distorts the architecture. So this is sort of where our understanding, at least our model, is that we think that the origin of human metaplastic basal cells comes from the endogenous stem cells that is lost during the fibrotic process, and that the fibroblasts themselves can drive this pathologic signaling. So then, relevant to a therapeutic paradigm, what you want to know is this basal cell metaplasia actually reversible? And so this is work, I'm going to talk about work from a very talented postdoc in my lab, Tsukasa Kadota, as well as in collaboration with our technician, Rose Zhao. And what he did was that he wanted to know, well, what happens to these metaplastic basal cells afterwards, right? After they come around, what we want to know is during maybe the injury resolution phase, what is happening to these basal cells? So employing a technique where we can genetically label these abnormal basal cells after injury. What he could do is he could follow them out as after the injury process, as the lungs start to slowly try to resolve some of these injurious processes. And what he found was that as you sort of follow these basal cells out, most of them stay as basal cells, but a very small population of these basal cells eventually becomes something that looks like an alveolar stem cells. But this happens actually not very efficiently, as you could see, by the number of those cells that eventually becomes alveolar stem cells. So there appears to be the lung appears to have some sort of endogenous reparative mechanism to turn some of these basal cells back into the endogenous stem cell of the alveoli, but this doesn't occur very efficiently. So the question then we have is, well, can we learn something about the microenvironment of those stem cells to think about how we can drive and make that process more efficient? So this is another work from our lab where we previously demonstrated that at least in the mouse, when during the fibrotic process, when these progenitors are turning into metaplastic basal cells, if you turn on hedgehog signaling in the scar forming fibroblasts, that will lead to some sort of local environmental cues to drive the formation of these metaplastic basal cells. Again, all this is supposed to tell you is that fibroblasts can be a very critical factor in driving this metaplastic basal cell transformation. But what we wanted to know is what happens afterwards, right? So that's the initial injury, but what happens after in the late injury process? So here, what Tsukasa did was that he took basically early and late injury lungs after fibrosis. And what you just wanted to look was to see, well, what are the fibroblast populations present? And again, using this technology called single-cell RNA-seq, what we can do is we can actually look at the full breadth of the types of fibroblasts that are present. And the take-home point from this slide is just that, during early injury in the bottom of the slide in these blue cells, what you have is a bunch of fibroblast cell types that we know are there in response to fibrotic injury, those that are inflammatory, those that produce collagen. But long after, actually, during the resolution phase, you get this reemergence actually of these alveolar fibroblasts that were previously there, but lost during the injury process. So you have this reemergence of a population that actually was there during homeostasis. But what I'll say is that they're not quite exactly the same. What we found was that when we looked a little bit more closely at these reemerged alveolar fibroblasts, is that they start to express secretory factors that were not there before. And one of them was a factor called HIP. And HIP is called hedgehog interacting protein. And what it is it actually an extracellular antagonist. It's a membrane tether protein with an extracellular face. And the only job that we know of what it does is it acts as an extracellular decoy to soak up the hedgehog ligand and prevents basically the activation of the downstream pathway. And for some reason, your lung, after this fibrotic injury, decides that it's important for the alveolar fibroblasts to express it, and they were not there before, okay? So, and what's interesting about the localization of these de novo HIP-positive alveolar fibroblasts is that they tend to be in areas where you have resolving injury. They tend to be an area where you have endogenous alveolar type 2 cells, and more or less restricted from areas where you still have a lot of injury with metaplastic basal cells present, suggesting, again, they have a role in the natural resolution of that injury process. So the question then posed to us then is, well, okay, if nature has this sort of endogenous reparative mechanism, it's not so efficient, how can we make it more efficient? So the thing that, so here's the question, right? Can we actually reverse some of that process by making this process more efficient? And so what we did then was we generated a chimeric protein, knowing the bioactive domain of HIP. What we could do is actually link it to the FC region of an antibody. And this is really the backbone of almost most of the biologic therapy that you have heard of out there today. And we sort of engineered it this way, this protein called HIP-FC. And what we did was it gave this protein after the induction of basal cell metaplasia in our fibrotic model while we followed the fate of these metaplastic basal cells treated with HIP-FC. And what we could see is that when you give HIP-FC, we got more type 2 cells. So we were able to follow the fate of these metaplastic basal cells. And as they sort of starting to emerge from injury, HIP-FC was able to increase the differentiation of the basal cells back to the endogenous alveolar stem cells. And what this also did was it improved some important physiologic parameters that you might care about, including the fibrotic burden of the lung, as well as the oxygen saturation or the physiology of the lung to sort of exchange a gas. Now I should congratulate myself. Well, I cured pulmonary fibrosis in a mouse, right? This clearly will lead to a very important therapy or not. So one of the major barrier of any sort of lung research or specifically pulmonary fibrosis research is that the model for lung fibrosis is not great, right? Bleomycin doesn't really recapitulate a lot of the aspects of what Paul described in IPF. It's very complex. So the goal that we set for ourselves is trying to sort of engineer a model that's going to be more like that of human IPF that is amenable to a drug treatment. So what Tsukasa did was he basically established a humanized model of metaplastic basal cell differentiation. So what he did was, again, he took the human alveolar type 2 cells that we talked about, that is the cell of origin for these basal cells in IPF lung. And he basically transformed them into basal cell in vitro, expanded it. And then what he did was he adoptively transferred them into a immunodeficient host that's injured, so to allow for engraftment. And it's sort of similar in a way to a lot of the PDX model that you might have heard of in the ONC model. But in this case, we're taking an epithelial stem cell that is normally found in the fibrotic lung. And again, reconstituting it into an injured immunodeficient host. And what you can see here is actually the appearance of some cystic lesions that you see in IPF lung. And I would say one of the biggest complaint about the bleomycin injury model in mouse is the lack of this sort of cystic structure that you see that recapitulated in human IPF. And what we can see again here is that this is the honeycombing that you see. If you're a resident and you've been on pulmonary consult with us, and we would take you to the CT reading room. This is all we talked about when we look at a CT and fibrosis. Is there honeycombing? Is there honeycombing? Is there honeycombing? This is what it looks like under normal pathology, right? Is these sort of appearance of these cystic spaces that are stacked on top of each other. And what the cystic space is going to have is actually going to be lined with cells of airway epithelial origin. So in this case, basal cells, secretory cells, and such. And that's what we see in our humanized model. We see these cystic spaces. Again, the NUMA actually give us an ability to trace cells that are of human origin in mouse. And you see all the green cells came from a human, forming this beautiful cyst with both basal and secretory cells lined within it. So now we have a model to actually trying to understand whether our compound is able to reverse some of this process. And the answer is yes. So we utilize this model to treat with the HFFC. And what we can see is that there seems to be actually significant reduction in the proportion of the transplanted cells of basal origin, while an increase in those that actually gain type 2 cell differentiation. And then we can also see that there's maybe also some improvements in their physiology. So this sort of sums up our sort of therapeutic sort of paradigm in terms of how we think about going from some basic understanding of the epithelial dysfunction that occurs in this fibrotic lung disease to how we can think about how we can try to come up with ways the body normally tries to resolve injury and try to sort of push that along a little bit. So the working hypothesis that we have so far, that Tsukasa is working with, is that there is this endogenous emergence of alveolar fibroblasts that maybe have some reparative function to resolve this basal cell metaplasia and what you see, which is basically the formation of these cystic space, bronchioli space that are honeycombs, to try to sort of turn it back. It's not very efficient. And perhaps there is a way we can do to sort of make a sort of a pharmacotherapeutic approach and make that more efficient. And that is sort of currently where we are at. And with that, I would again like to thank Tsukasa as well as Rose, who did most of these studies. And again, with that, Paul and I will take your questions.
Great. Why don't you both come back up? Paul, is the telomere mechanism, is that lead to fibrosis in other organs, or is it lung specific?
Yeah, so they've modeled telomere dysfunction in kidney and liver, and they get fibrosis there as well.
And do the patients with IPF have a risk for fibrosis in other organs?
Yeah, again, a third of IPF patients, if you look for it, will have cirrhosis. So the answer is yes.
And Tina, for those of us that are not in this line of work, so how does it work in your world as you are coming up with these potential therapies to go from your lab to potential commercialization, or kind of scaling that to potentially come up with a drug that might work?
Yeah, I don't know. Do you know anybody that can do that?
Is that just a matter of money?
Yes. I think this is where I think institutionally we can think of ways to connect investigators with biotech. And I think within UCSF, there are some mechanisms for that through the innovation office, as well as some other ventures that are joint, sort of trying to develop compounds within. UCSF actually is pretty good. We are currently sort of have a joint agreement with one such entity to try to see if we can develop this. But I think that's a really good question. I think that is I think it becomes a limit. Like, a lot of people at UCSF and labs have good ideas. How do they translate that into and find commercial partners? And I feel like there should be a better way.
Yeah, but just also in terms of when it's the right time, you feel like, it sounded like you've done a lot of the foundational research, and now you have some candidates that seem to be influencing fibrosis, and as you said, you're going to win the mouse Nobel Prize. When do you know that is the right time, that you have a candidate that really is viable to be a human drug?
When is it the right time to go to a company? It's when you know somebody. I mean, this is the problem. Again, I think this is what they're stochastic, right? Is that you don't, I will tell you that when I brought this to people, what they've said is like, wow, you have this really established. Most of the times we see this at a phase pre-development of the compound. But what people might have is platforms and things for discovery, right? And that's actually, it's really all about how you can sell.
Okay. And in terms of the immunosuppressive thing, I was brought up in an era where that's how you treated these diseases, and you looked on the scan, this was even maybe before scans, for evidence that there was an inflammatory component versus all fibrotic, but it was inflammatory. It was a no-brainer that you put them on immunosuppressives. First of all, are there any patients that benefit from immunosuppressives, and why do they not work in at least some potential, some group of these patients?
Yeah, I think, and I didn't really get into this, but there's sort of, IPF is just fibrotic and not inflammatory, to sort of oversimplify. These other diseases are autoimmune disease, hypersensitivity pneumonitis, are probably initiated by inflammation, broad term. And if you use those drugs early enough, you can favorably modulate the disease.
Mhm.
And then we're learning that they can transition to more of a fibrotic phase. And when they sort of transition, and when that transition happens, frankly, it's hard to show. But when they do, it's just like IPF, basically.
But it's a clinical decision. It's not so much that you would take someone who the scan and their PTs would tell you that maybe immunosuppressants work. But are you then doing genetics on them and saying, no, they won't in this patient because the telomeres are screwed up?
Yeah, these are great questions, because I mean, this is like the gray zone right now, Bob. And we're still using the clinical diagnosis as the main driver of treatment, but this short telomere data is provocative. We're starting to incorporate it. We don't have a great consolidated treatment pathway, frankly, but I think we'll be incorporating it more. The other thing that's being done clinically is that these quantitative lung fibrosis CT scans. People are trying to quantitate the fibrosis itself and use that as a predictor of transition, when you're more fibrotic and when you should be using antifibrotics versus immunomodulatory drugs.
All right, let me open it up. David, say you are.
David Earl. I was struck by the not just the ineffectiveness, but actually the harm caused by steroids in individuals with short telomeres. And I'm wondering if we know the mechanism for that, why it would be harmful, particularly in that group, and whether anybody has looked to see whether when we use steroids to treat other diseases, do short telomeres also predict a bad outcome?
Yeah, so David was asking whether using immunosuppression diseases other than pulmonary fibrosis, if a patient has short telomeres, is it harmful? And the first question was the mechanism. Yeah.
Generic, that you say are good for you.
Yeah.
Specific.
So David, we don't know the reason why they're harmful. We speculate that it's due to infections. Patients with short telomeres at higher risk for infection, just de novo. You see it post-transplant. These people have more CMV in post-transplant. So we speculate that they're at higher risk for infection, but again, that's speculation. And then I don't think anybody's looked at it. It's a great question. I don't think anyone's looked at it in other disease contexts that I'm aware of.
And higher risk for infection because you mentioned that they sometimes follow for fibrosis. There's something about the immune system that is specifically.
Yeah, so their bone marrow is sort of messed up, and there are specific elements of T cell subsets that are dysfunctional, so they're more prone to viral infections typically. Yeah.
Others? Yeah.
Prescott Woodruff, also pulmonary critical care. So I think simplistically about fibrosis on the one hand, but emphysema on the other as being the two archetypical outcomes of disease of aging in the lung, right? And actually both telomeres and HIP are important in COPD, potentially in different ways. So how do you see, could you speculate on what might drive disease in one direction or another? Or are these fundamental pathways in both diseases?
Well, here's what I'd say.
So
I guess it's telomere. So COPD was on that list, Prescott, for Mendelian randomization. It didn't get linked to COPD. But it has been, short telomeres have been found in patients with COPD. I think there's a strong gene environment interaction there, smoking primarily. And then in fibrosis patients, the gene part of it is more dominant and they get fibrosis. It's sort of how I think about it.
Yeah, that's something that we've often thought about. What I will say is that I sometimes maybe inaccurately look at emphysema and fibrosis as somewhat of opposite ends of the spectrum, and you can sort of and there are some genetic examples, like having too much TGF-beta can drive fibrosis, but too little can cause actually emphysema morphology in the lung. So in a way, they're sort of dichotomous outcomes, and maybe a bit of a Goldilocks effect is that in a normal lung, you need to have just enough of some of these. Too much of some can lead to one outcome, and too little of it can lead to the other, if that makes sense.
You ever see patients with both?
Yes, right. So we actually see it combined, but in different areas. There's a syndrome.
Singer.
Hey, John Singer, also from pulmonary. Tim, do you think there's a role for this compound, the TIP-FC, in potentially reducing bleomycin toxicity when used as chemotherapeutic regimens?
In humans, not in the mice.
In humans,
I'm
yeah.
very worried about.
Like with pulmonary toxicity.
So the question is can we use this to treat bleomycin-induced toxicity? Sure. So I think really, do we see a lot of bleomycin lung toxicity still? Is there a fair amount? To me, honestly, and I'm going to leave this maybe Paul to discuss, is whether that we see similar pathophysiologic findings in terms of bronchialization. But my understanding, again, some of these drug toxicity is more of an organizing pneumonia pattern. It might not arise actually to that degree of remodeling of what we're trying to target.
But just in terms of the way it works, would it be the kind of thing that you could use as a preventive if it were effective, as opposed to there's already damage that you're trying to resurrect?
If I'm the one selling you this drug, then I would say yes. But again, not knowing enough about the disease itself, I am
definitely pushing you to put on your marketing hat.
There's a patent.
I mean, all I'd add is, and Tian's right, in the early phase, it's mostly inflammation organization for sure. A subset do get durable fibrosis though, but I don't think anyone's looked to see if like bronchialization is an aspect of that remodeling.
Yeah, so 2 questions from the audience. One, and there's about 70 people watching online. One person asks, what are factors that we know that contribute to short telomeres? What can you do about it? And another person asks about how might ARDS and fibrosis after COVID be similar or different than IPF?
So what causes short telomeres? Genetics, we talked about that. Smoking, stress. Those are things that can shorten your telomeres. Aging, that's why IPF is an aging-related disease. Can anything reverse it? Exercise, I think. Exercise and good, happy living will help you maintain your telomeres. So
You heard it here.
that's right. And the second one was what again?
The COVID fibrosis
COVID.
versus IPF.
So we did a study with Carolyn Calfee looking at telomere length and risk for outcomes from acute lung injury, not COVID per se. And it was interesting. Short telomeres was not a risk for the onset or acquiring acute lung injury, but if you had acute lung injury, you did poorly. You had longer time on the ventilator and worse survival. So it seems you can't heal your lung as well if you have short telomeres.
And I can only answer that's the second part, which is the difference between COVID or severe, let's say severe viral pneumonia and IPF, is that we've seen, again, we've actually published this too, there you see some similar ectopic basal cells in the alveoli, both in COVID as well, but they're not quite structurally organized similarly to that of IPF. And part of that is just the rapidity of the remodeling difference between the two phenomenon, as well as the fact that there's a whole slew of also inflammatory infiltrates that you would find in COVID pneumonia or viral pneumonia, IRDS, versus that of IPF. So the two have some similar outcomes, but again, there's so much differences in their how they arrive there. The lung actually looks quite different.
Did you have a question? Yeah.
Sure. Allison Wong, pulmonary medicine. Bringing it back to the bench, the HHIP finding is so striking. I wonder, my understanding is there are other hedgehog inhibitors being investigated in cancer agents. I wonder if you've tested any of those small molecules in your secretory cell honeycombing model, and if you're able to recapitulate results.
Yeah, the question is, yeah, so there are two FDA approved smoothen agonist, one from Genentech, one from a company that I'm not aware of, but there are two of them. And there had been earlier efforts to actually look at them in fibrosis. And I don't think there was a lot of interest there. What I will say to you is that we have looked at it somewhat, and there have been some studies published in looking at targets and efforts to target smoothen as an intermediary of the pathway in bleomycin lung fibrosis model. And I'll tell you, they didn't work very well. And it partly might be due to the timing and where, but we have also realized as somebody that studies hedgehog pathways, that pathway is really complicated. So there are multiple levels of activation within that pathway, and smoothen is only one of them. HIP is going to target it at a more upstream pathway even before the signal relays to smoothen. And so I don't typically find the two as comparable strategies, but it's a really good point, actually, I will say, when you're talking to potential industry partners about it, because they will raise that issue of, there's this thing redundant, and I would argue that it's not. And we have actually data in other diseases where smoothen antagonists and HIP works actually have very different outcomes.
Last question, Paul. We have a lot of pulmonary folks here, but a fair number of people who are not. Can you, and this is hard in 3 minutes, but can you give us sort of a lay of the land in terms of therapies you talked about emerging drugs and emerging approaches, but there are some treatments now for IPF that didn't exist 10 years ago. Sort of how do you think about that group of treatments and how well they work?
Yeah, I didn't really talk about the IPF treatments, but there's two drugs, nintedanib and pirfenidone. And for neither drug, we know it's MOA, frankly. But what we do know is that it cuts progression in half. So those patients, instead of losing 300 milliliters of lung function a year, they'll lose 150 milliliters of lung function a year. And then after those successes, they tested it in other fibrotic diseases. And there's a clinical phenotype that's emerging in our field. It's called progressive pulmonary fibrosis. It's that transition phase that I was referring to. And the drugs seem to be beneficial once a person transitions from an inflammatory feature of their disease to more fibrotic. Mm-hmm. So that's the state of the art. They're really hard to take. They have a lot of side effects. So there's a lot of work to be done. And frankly, the work that Tien's talking about, really people are starting to focus on the epithelium, which I'm biased, but I think that's, it targets more of the root cause of the disease, and there's better opportunity for success, I think.
But in general, you saw an IPF patient today, you would be thinking about trying to get them on one of these therapies.
That's right.
Yeah. Okay, so it's very good. Thank you both so much.